Wow, Sandhya, that was such a lovely introduction. I hope I can live up to 10% of what you just heard. But I am so grateful to be here giving this lecture on marvelous molecular motors. And I hope over the course of the next hour, I can share with you why I think they should be called marvelous. But before I talk about the science, I also just want to express my gratitude for being invited for this distinguished lectureship series. And I want to offer my special recognition of TNQ's founder and the real person behind this lecture series, Mariam Ram. She is such a passionate supporter of the life sciences, and as you heard from her, really trying to stimulate the next generation of scientists in India. She is deeply passionate about this, and I think we all should be very grateful for the efforts that she puts into this. So thank you so much, Mariam. I haven't been back to India in 3 years. I did a sabbatical here for 9 months. I came back once or twice a year. And, as you've heard, I love being in India. I have so many wonderful friends and colleagues now in India. I see this great potential of science here. It's terrific talking to all the students that I meet in India. They are also so bright, so passionate, and it's a great pleasure for me to come to be involved in many of the efforts that you heard about from Sandhya. But one of the pleasures is actually, it's not just me making all of these projects, but it's working with Indian scientists like Sandhya and Shubha Tole, who were also, I should credit, very influential in starting the Young Investigator meeting. This is the first meeting that happened in 2008. This is the 10th, and the next meeting in 2024 will be 15 years. This is an article I wrote with Shubha many years ago. I wrote an article on Indian science with my wife, Karen Dell, and also started a microscopy course, and you heard about India bioscience. But, for me, these have been really labor of love, and they've been projects that I've tremendously enjoyed. So the science I'd like to tell you about today is about these amazing machines which are called molecular motors. And they are the driving force of all of the movement that you experience or see in biology. So for example, some of the obvious, motion is one of the most obvious attributes of living systems. It's obvious all around us. We can watch animals racing across fields. Movement is essential for the entire animal world. But also, when you look under a microscope, you can see that almost any piece of soil or water is teeming with moving cellular life as well. And if you look inside of a cell with a very powerful microscope, as I'll show you later, one can also see that the interior of a cell is filled with moving objects, material being transported from one part of the cell to the other. And in addition, even the process of cell division, the process of mitosis, involves physical motion as well. So what you can see here, for example, is the DNA, which is shown in green here. And the DNA physically separates to create two daughter cells. So part of the action of cell division, again, involves physical motion. So basically, everywhere you look in biology, you find that motion is critical for life, whether it's at the macroscopic level or at the microscopic level. So the study of cellular motility also dates way back. The Greeks actually were the first to develop theories about how muscles work and how animals move. And even in the cellular world of motility, the studies go back to the origin of the microscope. So this is Van Leeuwenhoek, and this was his first original microscope, or a replica of one. And what he saw when he put this to his eye in some pond water is that, to his amazement, he saw cellular life for the first time and all kinds of moving objects in a drop of pond water. And what he said here was, in one of his writings, the motion of most of them in the water was so swift and so various, upwards and downwards and roundabout, that I admit I could not but wonder at it. So you can capture, this sense of amazement when he first saw this kind of motion and cellular life, this invisible world first brought to life to mankind with this, or humankind with this microscope. The discovery of motion inside cells occurred about 100 years later with this scientist Bonaventura Corti. And when he looked in some plant cells, he saw chloroplasts, such as shown in this movie here, and he saw that these intracellular material was also moving inside of cells. And this is what he wrote. I know that the phenomena that I announce is too strange to be believed at first. I saw two torrents inside each section. One of the torrents rose on one side and descended on the other constantly. And this not once, but thousands of times, and for days and for entire weeks. It's too bad we don't write scientific papers like this anymore with this kind of enthusiasm. And what I think these also quotes really capture, which I want to also convey to the student audience here, is this amazing excitement of discovery. And this is why you do science. This is what the magic is about being a scientist. Seeing or understanding something new in the natural world. And what I also want to emphasize is that, of course, in these distinguished lectureship series, you get scientists who are a bit older and have made famous discoveries. But I remember, as Sandy was mentioning, when I was 10 years old in my basement, or when I was an undergraduate, and I was doing some research where I found out something, even if it was an experiment that revealed something new, it was an amazing feeling that I made that discovery, even if it's small, that no one else in the world ever knew before. And that is an amazing feeling. And that feeling was as wonderful to me when I was a child or a student as it is for me today. So I hope I can capture a little bit of this sense of the discovery and the excitement of it, and why that makes the life of a scientist so rewarding. Okay, so what I would like to do in this talk is to tell you 3 short stories about 3 different molecular motors. So I hope by the end you'll understand a little bit about how all of these molecular motors work, and a little bit of my connection to these 3 motors, which have evolved over my career. So the first is a story about myosin. And myosin is a motor involved with the movement of the cheetah that I told you about, or as I'll tell you in a second, the beating of your heart. And this will be a story of therapy and medicine. You heard a little bit about this from Sandhya, about a company that we founded based upon molecular motors. And I hope to illustrate here just to show you the medical importance of motor proteins, and also the importance of basic research, that when you understand how something works in great detail, you can then begin to think of ways to apply it, to the good of society. In the second story, we'll talk about this motor kinesin. And that will bring me back to a large trajectory of my career, ever since the discovery of kinesin to understanding how it works. And then towards the very end, I'll tell you about a third molecular motor that's called dynein. And here I'll just tell you a short story about some recent work that we're doing right now in the lab. Not that it's completely finished, but this will give you an idea of a snapshot of science in time, not just in the past. So this is the outline of my talk with you today. But let me tell you a little bit about what moleculars are before we dive into each individual one. Molecular motors are proteins, and they are proteins that can convert chemical energy into motion. So that basic process, you're familiar with this in the macroscopic world. That's how we get around in the world, or attempt to get around in the world in Mumbai as best we can. And that is the automobile engine. Now, Tata came up with this car that they called the Nano, meaning it was a very small car. But nano on the meter scale is very, very small, much smaller than the Tata Nano. So the engine of the Tata Nano may be about a meter in size, but these molecular motors really are nanoscale. So these motors I'll tell you about are on the order of 10 to 50 nanometers in size. So that's like about a millionth of a centimeter. So these machines are very, very small. Like a car, they also use chemical energy, and they convert that chemical energy into motion and work. So your car will take gasoline, which has hydrocarbons. The engine will break down those hydrocarbons. That energy will be used to propel the car. In the case of the motors, they use a chemical source called adenosine triphosphate, which many of you probably learned about. The speed at which these move is about a few millimeters per hour, which sounds, pretty pathetic compared to a car moving, except if you compare the relative scale of how fast these two engines are moving in relationship to their size. So the molecular motors, in fact, when you compare to the number of lengths that the motor is traveling per unit time, actually move faster than your car does on the highway. And if we had molecular motors propelling human beings around the world, we probably wouldn't have as many problems with global warming, because the work efficiency of these molecular machines is far better in terms of converting the theoretical energy from a chemical source into work than your gasoline engine. So we have actually a lot to learn about how these molecular machines work, even from an engineering point of view. Okay, so the other thing I need to tell you about molecular motors is, like your car, motors need roadways for movement. Of course, we're used to this. The density of vehicles is significantly lower than in India. But you can see that there are specific roadways, and the engines move along these roadways. Well, in the cell, there are also roadways for these molecular motors, and these are called cytoskeletal filaments. And you can see these act as long polymers within cells that provide tracks for these molecular motors. Okay, so the first molecular motor in my three stories that I'll tell you about is myosin. This is what the myosin motor looks like later. I'll explain a little bit more about what this motor does. But what you're seeing here is at atomic scale here. So this is very small. And the roadway that it works on is called an actin filament. And actin and myosin were actually first isolated even in the 19th century, and that's where the name myosin was given by William Kuhn many, many years ago. So it was the first discovered molecular motor. Now let me tell you a little bit about how this motor works in your contracting muscle. So your muscle, as you may know, consists of many fibers, individual fibers that make up a muscle unit. So when we look at one of those individual fibers and look at what it's made of, one can see that along the length of that fiber, there are a series of repeating units, millions of them, that are called sarcomeres. And if we look at the individual sarcomere here, and this is an image under the electron microscope, you can see these dark bands over here, and then some lighter bands on either side, and then a dark line again. These dark middle bands in the middle consist of the motor protein, myosin, along with interleafing the track, which is actin. These lighter regions on the side just consist of the track, actin. So how does this sarcomere relate to the actual contraction of your muscle? So here's what happens in muscle contraction. There's the myosin sitting in the middle here. And there the actin coming from these kind of end zone points that nucleate the formation of these actin filaments. And what the myosins on this side are trying to do, they are pulling this actin filament towards the center, and these myosins are pulling these actin filaments in this direction. So if we look at over time, these actin filaments are far apart, and this zone is very large. But as contraction goes on and the motor pulls these actin filaments in both directions, this basic unit of the sarcomere shortens. And that is what happens when your muscle contracts and your muscle shortens, is that millions of these individual sarcomeres are shortening their size when these motors are pulling in these actin filaments. So let me now show you this in a movie, just to bring this into animation, in the case of your heart. So here is your heart beating. And now if we zoom in at a molecular level, as you'll see, here are the actin filaments here, and there are the myosin motors. And these periodically, when your heart contracts, the myosin pulls these actin filaments towards the center, and that is the contraction of a heartbeat. And then the motors relax, and then the heart distends again. So this periodic contraction, or the pulling of the actin filaments, is what gives rise to the contraction of your heart. So now let's zoom in on the machine itself, the myosin, and try to look at what it's doing. So first of all, I told you that this molecular motor uses chemical energy as its fuel. And kind of like your car engine, it breaks down this chemical energy. So initially, I was going to show that in a second. The motor starts by binding this adenosine triphosphate. It then cleaves a chemical bond between the second and third phosphate. That's what this is, adenosine diphosphate, and a cleaved phosphate group. And then the motor releases the phosphate, then it releases the ADP, and then it starts the cycle all over again. So this is almost like one stroke of the piston. And what does the motor do during this chemical cycle is shown in this video. It actually undergoes a choreographic shape change that causes its action to pull the actin and slide the actin as I showed you in the last slide. So here it is. The myosin releases this phosphate. That's this step. And that causes it to undergo this big swing, this big stroke that slides the actin filament a short distance of about 10 nanometers. ATP comes in, hydrolyzes it, and it recocks the motor for another stroke. So billions and billions and billions of these events all together in your heart is what causes it to contract. So even though the individual myosin movement on the actin is very small, when you put billions of these together, you generate a macroscopic contraction. So now that we understand myosin, and this is not work from my lab, but from hundreds of labs that really over time developed this very detailed understanding of what myosin does, one can then, with that knowledge, ask this question: can one develop ways to manipulate these motors in a way that could benefit human health? So this was done by a company, Cytokinetics. I'll show you that in the next slide. But one of the questions that they ask is, can one make a drug that activates the motor, actually makes it work better, more efficiently, more strokes per unit time, in a way that will cause a more robust contraction of the heart? Now, this was a big unknown undertaking when we did that, because most drugs that you will find out there inhibit their target. They don't activate it. It's a lot harder to make actually a biological machine work better than is found normally in nature. So we had no idea whether this was even possible. And that's another thing about science, is you have to sometimes take bold chances and aspire for projects that you may not think are even possible, but with the knowledge that we had in myosin, we felt that this was at least worth trying to do. So that was the foundation of this company. Myself, as a disclosure, I'm a co-founder of Cytokinetics, along with Jim Spudich, James Sabry, and Larry Goldstein. And what you heard a little bit of this from Sandhya was a very ambitious undertaking to first, purify myosin and a lot of the actin components in a test tube environment where you could test many, many drugs, hundreds of thousands of drugs, and test one by one whether any of these drugs was able to have this positive activating effect on myosin. So this kind of screen would not be possible without detailed biochemical and mechanistic knowledge of how this motor works. So that's the first thing I want to say. There's a big push these days towards pragmatic outcomes, but pragmatic outcomes are built on a lot of basic, fundamental, foundational knowledge about how living systems work. And without that basic knowledge, you really can't jump ahead to practical outcomes. So it started here. And then some early hits, meaning we found some kind of compound that looks like it did activate. It's still not a drug yet. You then have to get chemists involved over years to improve the potency of that drug by making new variations. You then have to test these compounds and improve the way the compound behaves in animal models. That can also take years. But at the very end of the day comes this compound, omecamtiv mecarbil. And so this is a real journey from biochemistry to animal models to eventually patients, as I'll show you. And what this drug does is illustrated by that movie that I just showed you for myosin. Remember this event that I showed you where the myosin touches the track, the phosphate comes out of the site, and that triggers this stroke that causes the sliding of the actin filament. Remember that? That is the force-generating step of the motor. And what this omecamtiv drug does is, this is a drug concentration curve on the bottom, and on the top is the speed at which this phosphate comes out and the power stroke occurs of the myosin. And when you add this drug, you can see that the speed of that phosphate release and the power stroke accelerates by like 4 to 5-fold. So it would be as if I replayed that movie, but replayed it 4 to 5 times faster. And that means in the limited time of when your myosins are active during a heart contraction, which are signaled by electrical conductance, more myosins are producing more strokes per unit time. And that means more contraction during the limited time when the myosins are active during the heartbeat. And what this looks like now in a patient, what you're going to see on the left is an echocardiogram. There is the ventricle and atrium here. And this patient has a disease which is heart failure, meaning the contraction of the heart is not as good as it should be. So on the case of the left, you can see this valve. And this valve is kind of fluttering. And the reason that it's fluttering is that the contraction of the heart is not occurring normally. And that it gives rise to this problem that actually leads to hospitalization and many deaths around the world, heart failure. You can see with this drug, you can now see the contraction happening much more vigorously, which causes this valve to kind of pop up. That is what the contraction of the heart should look like. And this is the same patient several hours after taking the drug. So we're now going to, this is now in front of the FDA in the coming spring. So we'll see if this now completes its journey to a drug that will be sold and be available for patients in hospitals. So again, this is the message that I came to. I also mentioned is that all of this drug development would not have been possible without this foundation of curiosity-driven basic research. That is the foundation, for really all kinds of new medical breakthroughs that we may want to make. All right, so now let me get to the second story of Kinesins. So this is kind of more my journey, even from the years of being a graduate student. So I'd like to share this with you right now. So the problem that I got interested in 1983 is this problem here. How is material transported inside of nerve cells? Okay, why nerve cells? I will say that transport occurs in all cells of the body, but this problem seemed to be most interesting to study initially in the nervous system, and that is because nerve cells are kind of the largest dimension cells in your body. So even one nerve cell, for example, the cell body, which is where your nucleus is, where your DNA is, it may be in your, it would be located in your spinal cord. And then it extends a long tube, which is called the axon. This also carries an electrical current to the very end of the nerve cell, which is the nerve terminal, which may then interact with a muscle and activate a muscle, for example, in your foot. So even though the cell body is only, so this is, the axon is really effectively what makes up the nerve that travels down your leg, which you're very familiar with. It's bundles of, thousands of nerves. And then the ending could be in your foot. So the cell body is very small. It's only about 30 microns, so it's microscopic. You can't see it. But the length of the nerve is, extends, could be over a meter in a person. So all the material that the nerve makes, the neuron makes, the proteins, the lipids, all of these materials, the building blocks, they're all made in this cell body here. And this nerve has to last you for your entire life. And that means that building blocks that are found in the nerve terminal don't last for your life. The proteins turn over, the lipids turn over. They have to be constantly replenished by the cell body. So these building blocks are made here in your spinal cord. They then have to be packaged and sent down this nerve to your foot. And the question we wanted to ask is, at the time, we knew that there was a transport system. We didn't know how it worked. Okay, first I want to give you an idea of the scale of transport, because I think it's a little bit hard to relate. It's always easier to relate to things, in the macroscopic world. So instead of this 30 micron cell body, let's imagine one that's 5 meters. So maybe that's between these two podiums. And this would be the cell body. It's nice round. It looks like the cell body here. I might be the DNA over here, be about the right size of the DNA. And then you can ask, okay, where's the nerve terminal from this size cell body? So I'll let you think about that in your mind for a second. Just imagine where, from this auditorium, where would the end of the nerve cell be? You don't have to answer right now, but just think about it. Pick a geographic location around here. All right, you all done with your exercise here? Okay. The answer is the nerve terminal would be in Pune. So from this stage here, there would be little, also like transport containers. Many of them may be about the size of this slide changer. And they would be transported from this auditorium all the way down to Pune. And that would take, on this scale, it would take about 2 days for this transporter to reach Pune. So that's the scale of transport. So this, that was the process, we're trying to understand. And in biology, the other lesson for students is it's very important to choose the right model organism for a particular biological problem. So in this case, rather than, well, first of all, no humans want to donate their nerves to study. But even compared to other animal models, we chose the squid for studying this process. And the reason for that is it has a very unusual nerve cell that I'll explain in the next slide. But we did all these experiments at the Marine Biological Lab. It's a beautiful place. If you ever have a chance to visit it or take a course there, I highly recommend it. And we went there because there were fishing boats that leave from Woods Hole and collect squid on a daily basis. So that's where we were. And the reason why we came, or I came from Stanford to Woods Hole, to the Marine Biological Lab, for the squid is for its giant axon. So it has a particular axon which is involved in the escape response for the squid. If you see it squirting out water and suddenly moving fast, that's because of this giant axon that's involved in innervating the muscle. So that's this axon here. And you can see how giant it is. This is a cross-section, but the axons in, for example, a human body would be about the size of the other axons that you see in the surrounding here. So this axon is really like the king of all axons in size. And that made it a very good model because we could take that axon out of the squid, and that's me dissecting this axon out of the squid. And you can now study this individual axon that you see in this picture over here and try to study the transport process in it. So the other big thing that happened at the MBL is that now what you're looking at in this movie is a microscope that is now peering in to the inside of that squid giant axon. This was a movie made by Robert Allen, Scott Brady, and Ray Lassick just before came to Woods Hole, and it was a really transformational, scientific work, and it had a great influence on me because here you could see this transport process in action. And this was made for the first time by putting a video camera on a microscope. So instead of looking at the microscope through your eyepiece, you can now use a camera and also, at that time, a rather primitive computer to increase the contrast. And again, almost like the modern Leeuwenhoek, could see things inside of cells that were never seen before. So that's what the transport inside the squid giant axon looks like. Okay, and there's me a few years younger. Haven't changed, right? And my colleagues, it's just the mustache. I think that's the only difference. My colleagues, Bruce Schnapp, Tom Rees, and Mike Sheets, who are my collaborators on this project. And what we wanted to know is in this video that you see. And by the way, these big, long, snaky things here are mitochondria. Probably have learned about mitochondria. These are mitochondria moving. And then there are a whole bunch of small vesicles that you can see in this movie, which are transport vesicles. And Sandhya does a lot of work here on understanding these transport vesicles and how they move in the worm C. elegans. OK, so we wanted to know what were the motors and what were the roadways. So the first step in this is we wanted to study the transport system out of the cell and in a test tube. Why do that? Well, in a cell, there's limited ways you can manipulate the system. You can watch it, but especially at the time without CRISPR and other genetic manipulations, it was hard to do experiments on it. So what we wanted to do is take the transport system out of, not only out of the animal, but out of the actual boundary of the cell, out of the membrane, and get the transport system to work in vitro. So the first step is you can take that axon, cut one end of it, and you can squeeze out the inside just like a tube of toothpaste. I don't have a good picture of squeezing out the inside of the axoplasm, but it kind of looks like this without the striped colors on it. But you can just squeeze out the inside of the axoplasm. And what you do, we found conditions where we could keep that transport system working. The transport system here didn't even know it was outside of a cell, but we found the right conditions where we could keep it going. So you can see all these little vesicles here moving along these tracks. These big snake-like things are the mitochondria. And so this was the first step in the process. All right. So we went into this, as many scientists do. You come in with an hypothesis of how something may work. And that's good. The only problem with that is hypotheses are usually based upon something that you know, because it's very hard to make a hypothesis about something that you don't know. So our hypothesis coming into it was that actin and myosin, the motor and the roadway that were also powering muscle, were also known to be found in other cells. And we thought that myosin may be, and actin may be the transport system in the axon. So we did this experiment of taking a myosin-coated bead and adding it to that axoplasm mix. And if those little filaments that you saw in the last video had some actin in it, we thought that we should see those myosins streaming along those filaments just like the native organelles. So that was the experiment we wanted to do. It wouldn't prove it completely, but at least it would indicate whether we were on the right direction. So we add the myosin-coated beads to this axoplasm, and what we expected from our hypothesis, we would also see these plastic beads coated with myosin moving just like the native organelles. So that's what we expected. Now, the one other thing that students learn about, very important in science, is to do a control experiment. So that means you have a variable that you think is important, and you eliminate that variable, and then you should see a different result. So in this case, our hypothesis was the myosin was the motor, the bead was irrelevant. We'd see motion. So if we then had the beads, very small beads, without the myosin, we shouldn't expect anything, right? So that makes sense. Good control experiment. All right. So these were the results. Yeah, well, sometimes things just don't turn out as you expect. And the message is, rather than being disappointed by this is where the biggest opportunities in science come from. So my main point, and you'll hear this at a couple other points in this talk, is scientific research can be full of surprises. And this is something that to be treasured and also understood, because science is often confused with other disciplines like engineering and medicine. And in engineering and medicine, you do not want any surprises. if you're going over that bridge, you don't want to go, and if you're going in for, surgery, surprises are not, one of the things that you're looking forward to about the operation. But, surprises, but, in this case, there's a lot of knowledge here that's being applied. But, in the case of science, you're really at this forefront of the known and the unknown. So you're really venturing into lands that are unexplored. And it's part of the, joy of explanation to encounter new things that you don't expect. So this is the message I want to convey. The most glorious moments are when your pre-existing ideas and hypotheses that you may even have deeply treasured prove to be wrong or incomplete, because you're on route to discover something really new that even you didn't know about or anyone else knew about. And this is where the detective work really starts, and almost, I would say, where the most fun begins. Okay, so our initial hypothesis was wrong. We need a new approach. So we, it looked like actin wasn't the right roadway, so we needed to find out what the roadway was. And, we did this by doing actually a very difficult experiment. It took over 50 tries to get this experiment to work. And mainly Bruce Schnapp and Tom Rees were the really key drivers of this, but was to take this preparation live under a microscope and prepare it for electron microscopy, where you have much higher resolution and you can actually see the details of what these filaments look like. But we had to find the exact same filament on our video image as we found on the electron microscope, just to match them. And that proved to be very difficult. But we, thanks to Bruce and Tom, got this to work and found out that they were microtubules. And that is another kind of cytoskeletal system. It has this characteristic pattern of these repeating patterns of tubulin dimers, and you can see that repeating pattern in this EM, and also the diameter was correct. So we knew now what the roadways were. Now the question is how to find the motor. So, here, we really had to start from scratch of finding it. And how do we go about doing that? So an analogy I'll use here is to try to understand something. One way to do that is to take something apart and put it back together again. Maybe some of you had that experience of building radios, or maybe some of you are mechanics. But imagine, like, if you were like a Martian or something, came to Earth, you saw this thing, a car, and you really didn't know how to work. You'd start taking it apart, and you begin to figure out which of the parts that are essential for movement and which of the parts that are, not essential, right? And then you could try to take that car and put it back together again. And if you could take it apart and put it back together, and the car worked, you'd really think that you understood how a car works. So this was the same kind of approach. It's a process called reconstitution. It's led to many discoveries in biology. That's what I wanted to do. So the idea was to start with individual components that we purified, add them back together, and see if we can regenerate the transport process. So we knew how to make microtubules. So we can make purified microtubules. We could purify the membrane organelles from the squid. There was a procedure for doing that. And then we figured we'd add the microtubules back and the organelles, and we should get the system to work again if we added ATP. And the reason was, it makes sense that the motor would be attached to the membrane, because that's where the motor should be. It should be attached to the objects that it's moving, and we would be able to see the system work. So that was our assumption going into it, which a prelude is that assumptions can be wrong. So here are the experimental results. We had different tubes. We had the microtubules, the ATP, the membrane organelles. Combine them in another test tube, pipette it onto a slide, look at it under the microscope, waiting to celebrate. Finally got it all to work. Okay, bummer. Of course, try it again, same result, try it again, same result. And so then a negative result is often really hard because you never know whether you made some mistake when you made the preparation, maybe it was something you did. But you also have to keep the option open that maybe the idea of the experiment wasn't quite right. Maybe there's something else to look for. So to that, we added this combination of what are called soluble proteins. These are just proteins that are floating around in the cell. There are thousands of them. But we made an isolated tube of these soluble proteins, and we added all these things together to see if there was some missing component in this soluble protein mixture that may make the system come back to life. And yay, it worked. So we got videos that looked like this. So remember, going back to our control experiment, it's like leaving out a variable that you think is important. So we left out ATP, system didn't work, that's good, it needs chemical energy. Well, and then we did another experiment where we left out the membrane organelles, just to make sure that the moving objects weren't coming from the microtubule prep. That was the idea, that just to make sure that the organelles that we saw moving were really from the squid axon. So, this was supposed to be a 5-minute experiment. You finish it, you go home. Actually, it was around 1 in the morning when I was doing this experiment. And, everything was working well. So that's the thing. If things are working well, you just keep going. Didn't expect anything to happen. And then what happened under the microscope, I think the word mind-blowing is fair to use. We saw these microtubules moving on the glass. So there are no organelles here. Here's some maybe piece of dust or something, but it's a marker that's stationary. And if you look at all the microtubules next to it, they're sliding across the glass like little snakes. We did not expect this at all. So, okay, here's another thought experiment. You see that, what do you think? I'm going to pause for like 10, 20 seconds. Do you have an idea of why this would happen? So after, and this is like what we're doing. There was an initial shock, what the hell is going on here? But then, stop for a moment. Let's think about what's going. So maybe some of you have other guests. Some of you are puzzled. I'll tell you what's going on. And a good analogy here is crowd surfing. So this is at least one of my generational heroes, Bruce Springsteen. And you can see that the boss, Bruce, is traveling. He's being transported by people who themselves are not moving. They're stuck in their one spot because it's so crowded. But there are little arms here that are moving Bruce along the stage. And I thought, maybe that is what's happening. There are little arms that are stuck onto the glass surface, and those little arms, as shown here, that are stuck on the glass, are grabbing hold of the microtubule and moving that microtubule across the glass surface. So this meant that there were free-floating motors in this soluble material, not stuck to the organelle, but just motors floating around in the cytoplasm that would stick to the glass and move these microtubules. So that was transformational because now we had a way to find the motor. And there were maybe 10,000 different kinds of proteins in that soluble mix. And what you can do in this field of biochemistry is sort through 10,000 proteins to find the right one that would be responsible for that. So with biochemistry, you kind of create different ways of separating proteins, and then you test each of these separated buckets of proteins to see if they move. And then eventually, so this is a late stage of the purification. This is a gel. These are fractions of separated proteins at the end of a purification. And we've found like a new protein here. This is a protein here that was correlated with the presence of this motility. So this fraction had very little. This fraction had a lot. It corresponded to this protein, characterized the protein. It had no known, it was an unknown protein. And it didn't have any of the characteristics of myosin or dynein. So that was exciting. We're able to name that protein. My mom helped come up with the name, kinesin, and that's how the protein was named. So in addition to discovering the protein, this whole process allowed a new way to investigate biological motility. Because I showed you before that you can see the transport inside of a cell, but it was very difficult to study. It's a very complicated system, lots of things happening in here. But here is a very simple system of one protein interacting with its track with the chemical energy ATP. It could not be any simpler. And this was plastic beads. Remember I told you how the plastic beads moved in that strange experiment we couldn't explain? Well, these plastic beads picked up kinesin, and the kinesin moved these plastic beads along microtubules. So these assays became like the workhorse for understanding how this motor works. And this allowed us to really understand how something as small as 100 millionth of a meter, 1 millionth of a centimeter actually works. So this simple assay, in conjunction with other new tools that kind of came up from the investigation, ways of studying these motor proteins at the single molecule level, which I'll show you, and also getting their structures allowed us to understand how they work. So, for example, one of the single molecule assays that we developed, which I think Sandhya mentioned briefly, is we could then even eliminate this bead, get rid of it completely, and just have the pure motor with a fluorescent tag on it. First we used GFP. And now you're looking at a fluorescence microscope, and all of these individual spots you're seeing here are single proteins, single molecular motors that are moving on the track. And this allowed us to really begin to investigate kind of the characteristics of how these motor proteins moved and walked. And if you follow them in great detail, and there are ways of tracking this motion, you can see the motor actually not moving continuously, but moving in a series of like hops along this periodic track. So each of these hops is the motor moving from one unit here to the next along this track. So the question then became, how does it work? What's the molecular choreography? I showed you a molecular choreography for myosin, but we wanted to know the details of how this worked at an atomic level. So, with this group here, with Robert Frederick, John Kuhl, Elena Sabline, got the first crystal structures of kinesin. This is a space-filling model. This is often how structural biologists depict what proteins look like in terms of some characteristic features like helices and beta sheets. But this structure also brought a complete surprise. So surprise is the theme of this talk. But surprise was that before this atomic structure, everyone thought that kinesin and the myosin motor I shared with you were completely unrelated to one another. And there was a lot of reason to believe that. They moved on different filaments. They had different sizes from one another. Computer programs at the time said these motor proteins have no amino acid sequence similarity, and muscle myosin and the kinesin had very different motility properties. The myosin people went to their meetings, kinesin people went to their meetings. No one believed there was any relevance to the other motor until we found out that evolution had a different view on this. Both of these motors must have evolved from a common ancestor very deep in evolution. And you can see this. What you're seeing in blue here is kind of a common ancient structural core that is probably from an ancestor, protein ancestor that's now at least unknown to us right now. So the core, this is really the core engine that hydrolyzes the ATP. And that is a preserved core structure from actin and myosin. And kinesin and myosin. And kinesin learned to walk on microtubules. Myosin learned to walk on actin. But we deduced from this that there must be some like common mechanistic features. And that helped us a lot in investigating what the motor protein does. Now, one thing I just showed you in that video, I showed you that big lever arm. Remember that, of how the myosin produces its stroke right here? It changes its shape. It undergoes this big swing. That is the mechanical event. So we looked at kinesin, and there was another element, but positioned in the same spot as the mechanical element of myosin. And we made the deduction or hypothesis that that would also be the mechanical element for kinesin as well. So in this case, the hypothesis turned out to be correct. And we did a lot of experiments to show that are now reflected in this animation that I'll show you. You have to take animations with a grain of salt, but this is based upon atomic structures. But here is this little mechanical element in red, and here's the key thing. When ATP comes in, it zippers up along the side of the motor. And this is the next thing. Here ATP comes in. There's a zippering that occurs. And that zippering swings the rear head to a forward side. So it walks a little bit like a person. The rear foot goes forward. The rear foot goes forward, passes the forward foot. And the molecular choreography of this machine is that the front head undergoes a mechanical event that pulls the rear head to a forward binding site, and that allows this machine to walk in a very regular manner along the microtubule. Okay, so now where are we now with kinesins? Well, there are many kinesins. We now know there's not just one. There are 45 different kinesins. And they're not just involved in axonal transport. They're involved in transport in all cells of the body. They're involved in cell division. And we've now discovered that almost everything in cell biology seems to use one of these molecular motors. So that's what's happened over time. There are many unfortunate individuals that have mutations in these motors that cause rather tragic neurological diseases in children. So, I hope that in the future we can also take our knowledge about these kinesin motors, like I shared with you with myosin, and begin to think of ways in which we can introduce new therapies for individuals with kinesin diseases as well. Okay, so I have a very short story. The closing time is 6? Okay. Anyway, I'll definitely be able to get through it by then. Closing short story is dyning. Dyning was discovered by Ian Gibbons several decades ago. After we began to understand kinesin, we shifted actually our research to dyning because it was much less understood. I had the great pleasure working with Ian for many years, a wonderful colleague who's passed away not that long ago. But the reason why dyning took the longest is that the size of dyning is mammoth compared to kinesin. Maybe that last slide of kinesin made it look big because I filled up the screen. But if I put them on the same scale, you can see that dyning is almost 10 times bigger than kinesin. So it's one of the most complicated machines produced by the genome. So we spent many years, well, let me just tell you what dyning does. Kinesin moves material in one direction from the cell body to the nerve terminal. Dyning moves objects in the opposite direction from the nerve terminal to the cell body. It is also propels all that movement that you saw from Leeuwenhoek, which is driven by cilia and flagella. It causes this waveform movement that causes microorganisms to swim. So dyning is responsible for that as well. And what our lab is now working on, and many other labs, is trying to understand how that motility machine of sperm and cilia and flagella work. And this machine is much more complicated than the transport mechanism that I showed you for kinesin. This is a cross-section of that long whip-like cilia structure that shows that it contains a ring of microtubules surrounded by a central pair. And this machine actually consists of hundreds of different proteins, all of which make up this very unusual motility structure. The dyneins, the motors here, extend from one microtubule to the next. And I'll show you in the next slide just briefly how they work. Remember how I said myosin slides actin filaments in the sarcomere? Dynein slides other microtubules in the axoneme. So these dyneins over here are sliding these microtubules across from it. These dyneins are sliding this microtubule across from it, etc. So if you have all of these motors trying to slide all these microtubules at once, you're not going to get anywhere. So the way this machine works is to regulate the motors, and we still know very little about how this actually works. So now I'm going to tell you about a problem we do not understand. And that is that dyneins on one side of the axoneme of these microtubules are active, and they produce sliding, and dyneins on the other side, then it switches. These microtubules are off, the dyneins on the other side are active, and when they're active on one side, they can cause this relative bending of microtubules that causes the bend. And that's the bend that you see in this movie over here, is kind of this asymmetric switching of microtubules sliding on one side and the other. Okay, so the question that I'll leave you with is how is this dyning controlled? We don't understand this, but there are these additional elements here that are called radiospokes that are somehow responsible for controlling the dyning activity on the two sides. Don't know how it works. They're going to come into this very last short story that is currently ongoing in the lab that a very talented postdoc, Jen, is doing in collaboration with David Agard. And he started with this very basic question, which is, why do different sperm swim differently from one another? And here I showed you this movie of sea urchin sperm, and you can see it's nice, symmetric, planar beating, perfectly like almost a sinusoid wave. But if you look at human sperm or mammalian sperm, it's like this kind of corkscrew, three-dimensional asymmetric activity. So how does this come about? How do you get different kinds of beating patterns? Well, Zhen's approach was to get a high-resolution structure. So this is done now with another kind of technique which is very hot right now, called cryo-EM tomography. And it allows you to see structures, even very complicated ones, and you can get, instead of these stick drawings, these very detailed, near atomic resolution information about what this machinery looks like and how all the proteins look in the axoneme. So this is what Zhen did for his work. And what he discovered, he didn't know what he would get by going into it, but what he discovered was that mammalian sperm have additional elements in their radial spoke structures, the blue, the red, and the orange elements, that are not found in the radial spoke structures that were described from other sperm and cilia. We don't know what the molecular identity of these proteins are, but they appeared to evolve somewhere between fish and mammals. Now, the other thing that he learned from this structure was that the distribution of these novel new mammalian structures are not symmetric along the 9 outer doublets. So in other sperm and cilia, all of the radial spokes look identical across all the outer doublets. But in mammals, these new structures are different in the different outer doublets. So for example, these two have none of these blue structures here. and the blue structures are found in the other cases. If you look at the red structure, it's missing from a couple outer doublets, present in another. And this orange element here seems to be very specific for two outer doublets. So what Jen has found is not only discovered new structures, but he also now has emerged with kind of a new hypothesis of what may cause the different beating between a sea urchin and a human sperm. And that is that mammals appear to have an asymmetric distribution of a new structure across the outer doublets. And our hypothesis is that this asymmetric distribution of these new structures are somehow responsible for this asymmetric three-dimensional beating of mammalian sperm. So, this is the way science goes. I told you about some very mature stories of how, we've worked for many years to get an answer, but here are really some new questions that we don't know the answers to. We don't know what proteins make up these new structures that I showed you. And I told you an hypothesis that their asymmetric distribution is responsible for the asymmetric beating. We don't know if that's true or not. That's something that we have to figure out. So in the last couple minutes, I'll just tell you about this other passion, and you heard about it already, that we should make whatever knowledge we produce in science freely available to everyone around the world, to small colleges in India, to small colleges in the US, every country. I think that's our responsibility. And I think knowledge is really one of the great things that humankind has done. It's something to be celebrated, and it's something to be shared openly across all citizens of the planet. So you heard about iBiology. That actually arose from a trip to India originally. But here, if you don't know about it and you're a student, go to the site and look it up. It's a way of hearing talks by scientists, from the comfort of your home, as long as you have the internet. I'm really excited about this new project for undergraduate education, which is a close collaboration with TNQ. And I think this is trying to address two issues. One is that textbooks are very costly, and most students, even in the US, can't afford textbooks. And the other thing is, honestly, they're boring. They're just sea of facts. And it's missing, I think what I try to share with you today, which is the excitement of what scientists do, how they struggle with problems, how you have to come up and find ways to do experiments to solve solutions. It's all the detective work. It's failure. It's success. It's all of the wonderful humanity and the beauty of human beings trying to do creative work. And we don't teach our students that. So this is what this project is trying to do. You can look at it on the web. You can hear this story of motility. You didn't have to be in this auditorium. You could be somewhere else and learn about it. Or you can hear about CRISPR and how that revolution started, through a story told by Jennifer Doudna, who made the key discovery. So I encourage you to have a look at it. It's still in evolution. Sometimes projects take many years, and you have to be patient. So I think this idea of revolutionizing how we teach biology, moving away from facts to creating some sense of wonder and understanding of what science is and what scientists do, that's going to take years to try to change the educational system, not even in India, but in the United States. But it's an important journey. I'm really excited to undertake it with TNQ, and I hope all of you, I hope there are ways I can work with many of you also to excite students about biology in India. I would love that. And I hope I also convey to the students what a wonderful career science is. I am so grateful to have had this opportunity, maybe become a first-generation college student, but, importantly, find something that I love, I'm passionate about, And there's so many elements of science that are wonderful. It's making the discoveries, but science is often also a very personal human endeavor. I want to share that as well. One of the great things about doing science is it's not a lone journey. You're in a group with many people. It's fun to interact with them. The lab is like a family. And with that, thank you so much.